Thank you very much, Christoph, for agreeing to give a Build a Cell seminar, and you can get started.
Okay. Thank you very much, Kate. And hello to everyone. And thank you very much for inviting me. I'm really excited to speak about people having a very good knowledge about synthetic biology, probably more than I am. So Kate asked me to introduce myself. I'll do that by giving a very quick CV of myself because it also fits the introduction to the topic. I studied at the ETH Zurich and I did my PhD there in the group of named Peter Dimroth, and there I worked on the ATP synthase, this fascinating rotary machine that all of us know. And I was working mechanistically on the enzyme, so I tried to understand which amino acids are important. But then, whenever my PhD got longer and I had the chance to actually stay there as a very early on group leader, and I wondered what actually drives this enzyme. And to answer this, of course, I had to switch to the dark side. And the dark side I found at Stockholm University, where I started working with cytochrome C oxidases. And you have to know that in this time it was very exciting to be in any of these fields, but the two fields were really not mixing each other. They were having separate sessions, so they wouldn't meet at conference too much. So it was really quite a switch to moving from the ATP synthase, where we only had protons, then to the oxidase, where we also had electrons. And I think ever since I tried to combine these two fields. I'm now back in Switzerland at the University of Bern since 10 years. And Bern, for those who have not seen, is the capital. It's a small city, roughly 150,000 citizens. And we have one of these classically European looking universities with about 10,000 students. This is the view from our department, which is, I think, quite nice. All these mountains are more than 4,000 meters in altitude. Unfortunately, only the view from our department is nice because the one to our department is a bit less nice. It's a classical block of concrete. Anyway, I'm there in the Department of Chemistry, Biochemistry and Pharmaceutical Sciences, working as a group leader. This is my group last week. It's quite big at the moment because we just got a bunch of new bachelor students, but typically we are in the range between 5 and 10 students, 10 co-workers, including master students. So what are we doing? I really come from membrane bioenergetics, so I don't think that I'm a freshly born synthetic biologist. And I am really interested in working with membrane protein. They fascinate me because they live in this different compartment of the cell in this oily membrane, and that's actually where everything gets together. Things I'm not going to talk about today is, for instance, a very cool enzyme called superoxide oxidase, of which we think it helps bacteria to infect the human body and maybe to survive in macrophages, so that's quite cool. We also have a project in collaboration with a startup where we look at the ATP synthase as a drug target in leishmania. And we also do some scaffolding for membrane protein reconstitution. You will see that we do a lot of this process called membrane protein reconstitution into liposomes. And to actually get this in a more structured way than we do it, we try to use some viral-like particle as a scaffold to get good incorporation. So what I'm going to talk about today is a little bit about the methodology of membrane protein reconstitution and bottom-up construction of these ATP synthesis modules, because very obviously synthetic cells will require ATP. It will be very hard without that. And I will also touch some of our work that we have been doing with giant unilamellar vesicles. To give you a short idea, there will be a short introduction, and then in the first part, I will mainly talk about this in vitro regeneration and talk also a little bit about the technical challenges that we are having, and I will then also talk about some very recent work where we drive ATP synthesis by electricity. And then in the second, much shorter part, I will then talk about the use of giant vesicles as cell models. So I keep this very brief. I'm sure everyone knows how important ATP is. ATP is a small molecule. We only have a few grams in our body, but we use it so often that we regenerate roughly our body weight every day. It can be produced via several pathways: amino acids, sugars, fatty acids. They are all can serve as an initial source of energy, and they then essentially the ATP will then be used a little bit similar like electricity to do a lot of biological work, including movement gradients, uh, food uptake. So I think I find it always very impressive when I read that about 60% of all the ATP in the brain or in the gut are just used to keep a membrane potential that allows the uptake of sugar to just keep a basic membrane potential. It works a little bit like a battery. It comes in two stages. ATP is the charged battery, and of course, the uncharged battery is ADP. So what separates them is this phosphate group. So to regenerate ATP from ADP, we have to find a way to attach this third phosphate to the ADP again. The cells does this in essentially two different ways, and the classical one, or well, the classical one, maybe the more ancient one, is the so-called substrate level phosphorylation. I don't know, do you see my pointer? That
We
would,
see the not the laser pointer, but the mouse, the black arrow.
yes, I think I switch maybe to the pointer. I'm sorry for this. I think it works better with this one. We start with a so-called high energy substrate, although this expression is not correct. But anyway, we have here, for instance, an example with phosphenyl pyruvate. This contains a phosphate group which can be transferred to the ADP, and we end up with a product, pyruvate, and the second product, ATP. This is a rather inefficient way in terms, but it's a very predictive way. So there are several parts in the body where this is found: glycolysis, TCA, cyclor, acetyl phosphate during fermentation pathways in bacteria. But what is really nice about it, this is a one-to-one coupling. So for one molecule of phosphenyl pyruvate, you get one molecule of ATP. So this is very predictable. Now, the drawback of this system is that you need actually an energy input that is larger than the energy required to make this phosphoanhydride bond for forming ATP. So that means you have to create these molecules, phosphenyl pyruvate, acetyl phosphate, and that is quite energy intense. The second drawback, maybe also in relation to synthetic biology application, is that you end up in product accumulation. You have to add a lot of substrate in the beginning, and you will eventually end up with a lot of product, in this case pyruvate. And if you don't want the product, you have to get rid of it. So the second, and I think more elegant way on doing it, is something that could be summoned in the term chemiosmotic coupling. This, of course, is based on this rotating machinery, the FO ATP synthase, and this is driven by the so-called electrochemical gradient, which is also termed the proton motive force. And this consists of a chemical component and an electrical component. And the nice thing about chemiosmotic coupling is that there is a large variety of energy inputs that can feed this proton motive force. So we know the redox reaction, as we find them in mitochondria. We have light-driven reactions in cyanobacteria and plants, and maybe a bit less known are so-called decarboxylation reactions. And what all these reactions are doing is they are driving ion transport across the membrane. And if you transport an ion across the membrane, you both create a chemical gradient, but you also create an electrical component because you transport a charge across the membrane. And in essence, it's this electrical component that typically has the majority of the enzyme stored. Now, what is nice about this system is that you don't need to start with an energy input that is energy richer than ATP, to put it in simple words. Instead, you can just stepwise add one proton after the other, and it doesn't matter how big your energy input is, as long as it's sufficient to pump one proton across the membrane, it's enough to build up the gradient. And if the gradient is big enough, then the ATP synthase will turn and make an additional ATP. There is several levels on how this can be regulated, and a pretty fascinating one is that the searing of the ATP synthase comes in different gears, like you have different gears in bicycles. So the larger the ring is, the easier it is for you to turn the wheel, and for the cell, it's easier to make ATP with less proton motive force. The advantage is because the majority of the things you're doing is transporting protons. So you typically, if you have a good regeneration system or light, you don't have any product accumulation. On the drawback is you need membrane proteins and you need proton-tight membranes. And with that comes also the caveat that these two processes can in principle be uncoupled by uncoupling molecules. And of course, they are not good because they break the efficiency. So can this be used in vitro? And yes, it can. It has actually been done 50 years ago by Racker and Stöckenius to prove that ATP synthase works with such a proton motive force. And what they took bacterial rhodopsin from Archaeum, Halobacterium, halobium, and put these purple membranes into the liposomes. And when you shine light on it, they will pump protons to the inside, and ATP will produced in this. So what we are doing is we are typically taking respiratory pumps, and they have the advantage that they are much faster and then proteorhodopsin, and you can reach higher rates. You need less protein, and so on. However, on the other hand, they might come with the drawback that the substrate is not as available as light. On the other hand, light might not be the ideal substrate if you want to keep your system in the dark. Just for completeness, there is also a third type on how to do this. And this is just by depositing protons on this side. This can happen via hydrogen oxidation or photoacids. However, this system pretty quickly comes to a halt because there is no feedback loop for the protons to equilibrate. So this is in essence what we are doing. And mostly we are playing with bacterial proteins. So we really purify these bacteria and then put them together in different combinations. And as a quick introduction, I show you here the respiratory chain of Escherichia coli. We have a complex one, which can use the NADH, which is a very central energy hub in the cell. And this can create a proton gradient by pumping protons, but even more importantly, it delivers its electron to regenerate the NAD plus pool to the membrane-bound electron mediator quinone, and this quinone can then drive the so-called cytochrome oxidase, which eventually will reduce oxygen to water. So this is really the classical respiration that we find in mitochondria, but that is existent in much more variation in the rest of the kingdom of life. What is then very universal is the ATP synthase, which chemiosmotically drives these protons back, and by rotating these machines produces the ATP. And we were in the beginning very much interested in trying to reduce the complexity of natural membranes so that we could better understand the mechanism of these enzymes. So we purified them and put them together. And just that everyone is on the same page, I have a very quick slide I stole from a PhD student. What we do in the lab is we express the protein in bacterial cells, we solubilize them, and we can affinity purify them. We create lipid vesicles, typically multilamellar, and then we make them unilamellar by extrusion. And what we then doing is typically we slightly destabilize the vesicles by adding detergent. This does not break them, they keep the shape. However, they are tentatively ready to take up some of these membrane proteins. And we then do some tricks and remove the detergent. Eventually, we end up with this thing called proteoliposomes. And you will see many of those during my talk. And it all started when we did this experiment where we took the cytochrome or the quinol oxidase from E. coli, and we co-reconstituted it together with the ATP synthase, and this allowed us to drive ATP for a very long time in a linear way. So in essence, the system never stops as long as there is enough oxygen and as there is enough electrons to feed the BO3 oxidase. We started doing that by a synthetic electron donor in form of DTT. You might know that for reducing your proteins before you put them on the gel. And as an electron mediator, we use this short chain ubiquinone. This was fabulous for inhibitor studies, and we have used it a lot. It gives us the ability to play with the stoichiometries of the two enzymes. We can also change the lipid composition. Challenges is still finding experiment conditions where all enzymes go into the same liposome with a high efficiency. But even worse is that they tend to not go into these liposomes in a controlled, oriented way. And this is now something I'd like to briefly touch with two examples. In the meantime, we have a set of substrate with which we are able to drive ATP synthesis. So this is succinate, NADPH, light, and NADH as well. And maybe notably also air, which is a very recent achievement where we used a high affinity transhydrogenase that is actually able to extract the hydrogen from air and capture these electrons and feed it into the membrane. So we indeed had a solution ready, which you could just put on the table, and it would produce air from the reaction of the hydrogen in the air and the oxygen. So that was quite impressive. So why does orientation matter? And again, I stole this movie from my student. And when we think on how we want to orient enzymes, how we want to reconstitute, of course, we always draw the enzymes in the way we want them. So in this case, the oxidase pumps protons to the inside, and the ATPase has the head on the outside, so that the proton flow from here to there will create ATP. However, it is also possible that the ATP synthase might be wrongly oriented, and then all the pumping doesn't help because the ATP will never get into contact with the outside where all the ADP is that we want to upgrade to ATP. So this is not too bad because then we will eventually have also some that have the ATP synthase on the outside. What is even worse is if the oxidase is wrongly oriented, because then since the enzyme is activated by the membrane-bound quinone, both orientation will be activated. So it doesn't necessarily mean that only one population works, but if we have the quinol here in the membrane, there will be some that pump to the inside and some pump to the outside. Now, if you only have one molecule of these in the liposomes, you might think, okay, then you have different population, not so bad. But of course, we want to have several of these pumps in the liposomes to make the proton motive force creation more efficient. So the problem ends up if you have more than one of these enzymes in the membrane and they tentatively orient the wrong way, in the end, you will have always more that pump to the outside than to the inside. And since both populations are activated, in the end, you will have no ATP production at all. So it has been an interest of our lab to somehow control this orientation for quite a long time. And I'd like to briefly describe two pathways that we have recently published. The procedure to get there took a bit longer. So the first example is proterodopsin. Proterodopsin is like a relative to bacteriorhodopsin, but unlike bacteriorhodopsin, which is kind enough to spontaneously insert into a liposome and pump protons to the inside, proterodopsin does the opposite. It pumps to the outside if you just spontaneously put it into liposome. If 50 years ago Ephraim Rucker only had proterodopsin, we probably would have waited a bit longer until we would have believed in chemiosmosis. So we then thought, okay, how could we possibly test that? And we knew from studies of ourselves where we saw that the ATP synthase seems to preferentially orient with the large headpiece on the outside, and studies of fusion proteins by the Fotiadis lab and with proterodopsin, it looked like putting a big ball and chain onto proteorhodopsin would eventually lead to a reconstitution where this ball and chain stay to the outside of the liposomes. And we were then thinking, how should this look like? And this is the final product that we were having. So this is the protein, the proteorhodopsin. This is roughly 30 kilodalton. And what we put on top is this fusion protein that is, in essence, a maltose binding protein, which is a highly soluble protein. The question is now how to put these two together. And we have done it in a covalent way, but we also have done it in a non-covalent way, which makes the system a little bit more flexible. And this is what I'm trying briefly to explain to you. So we were thinking maybe we can make use of the his tag of the protein, because in proteorhodopsin, our his tag is on this side, and this is actually the side that we want to have on the outside. So we were wondering how could we possibly put this ball and chain and connect it to the his tag that it doesn't fall off but can be removed later. And we came up by synthesizing this chain here, and this chain essentially is this molecule here. It consists of a short peptide that would eventually bind to the maltose binding protein via spike tag, spike catcher technology. But on the other side has this tris NTA moiety. So all of these moieties are nitrilotriacetic acid moieties that we know from histag purification, but there are three of them in a very confined stoichiometry. And people have found that if you make these type of arrangements, the affinity of these molecules towards a histag is about 1,000 times higher than a classical NTA column. So in other words, if you just put these 1 to 1 or maybe 1 to 5 into solution, they will form a non-covalent but rather strong bond. And so we thought if we put these together and we make this ball and chain arrangement, then the tris NTA of the chain can bind to the C terminus of the proteorhodopsin, and we will eventually be able to reconstitute it with the chain, with the ball on the outside. And if we add histidine, the ball will fall off, and in essence, we have the protein oriented in the correct way. We tested this by just incorporating proteorhodopsin and having a pH sensor so that we could determine the direction of pumping, and we shone light on top of it and followed this spectrophotometrically. And as you can see, indeed, the black trace shows when we have no orientation, then there we observe outward pumping. But as soon as we have put that ball and chain onto it, either in a covalent way or in this non-covalent way I was talking about, the directionality of the pumping was reversed. We then added into those same liposomes also an ATP synthase and drove ATP synthesis, and as you can see, the outcome was the same. We had very little ATP synthesis if we did not orient the enzyme, but we got a much better ATP yield if we oriented proteorhodopsin by one of these two strategies. And since this histag is in principle a general method, we also used it for another enzyme. And recently in this work with the hydrogenase, we were actually able to also orient another enzyme with this method. Unfortunately, however, it did not work for our PET system. And our PET system is the one that I described already with the BO3 oxidase and the ATP synthase. But instead of using this synthetic electron donor DTT and the short chain quinone Q1, we wanted to use the physiological substrate. So we embedded the quinone into the membrane, and to give the electron to those quinone, we also needed a membrane. And we settled for this protein called NDH2. It's a NADH quinone oxidoreductase, so it doesn't pump proton, but it just takes the electrons from NADH and brings them to the membrane. And unfortunately, or let's put it like that, in principle this worked, but we found out it didn't work as well as expected, and we couldn't possibly understand in the beginning why this is the case. So, and in this graph, maybe we can focus on the green trace first. And the green trace is when we use synthetic electron donors, the DTTQ1. And you see there is a strong dependence on the lipids. So here we have the percentage of DOPC, which is our basic lipid for the liposomes. And we mixed the negatively charged lipid DOPG into these membranes. And we see that it gets worse and worse when we have more DOPG. So naturally, our liposomes typically contained 100% DOPC or just phosphatidylcholine. The DO is not so important. And then we saw the best collaboration of these two enzymes. However, if we then added NDH2, we saw there was essentially no activity left. And the reason for this is because NDH2 requires a negatively charged surface to attach. The attachment of this peripheral membrane protein takes place via an um, amphipathic helix, which is both hydrophobic and also positively charged. So if there is not if this electrostatic interaction is not taking place, then NDH2 will not bind and no electrons will be moved to the membrane. So we were thinking, how could we solve this possible conundrum? And first of all, what was the reason for this behavior? Why would this system here not work well if we had negatively charged lipids present? So we then established a method to determine the orientation by a fluorescent method. And to keep long story short, what we found is that the majority of enzymes were oriented in a way pumping protons to the inside, which is the desirable orientation if we only had PC lipids. But as soon as we added in negatively charged lipids like PG or PS, which stands for phosphatidylglycerol, the majority would pump to the outside, and we exactly would land in this situation where there was no ATP synthesis activity left. We concluded it might then be the charge, and to verify that, we also made liposomes that would contain positively charged lipids. They are, of course, not natural, but as you can see, we were almost able to achieve 100% of correct orientation of the AA3 oxidase. So we concluded the orientation is strongly influenced by the charge of the lipids used, and negatively lipids, which we would require for NDH2, is bad for ATP synthesis. And we confirmed this by testing also different concentrations of these positively charged lipids. And we found that there is a sweet spot where we can improve our classical BO3 ATP synthase system by just using a little bit of positively charged lipids, but at higher concentration. The orientation is still perfect, is getting better and better, but the overall system breaks probably because to interactions of positively charged lipids and ATP synthase. So yes, this leaves us with the conundrum. We would require positively charged liposomes for reconstitution, but we require negatively charged ones for measuring. So how could we do that? And we then came up with the following strategy. And this is just the head group of lipids that we have used. So this is this positively charged lipid called TAP. So this is a 3-methylamine. So you see here, this is constantly positively charged. But if you remove one of the methylamines, you get a protonatable group, a secondary amine. And luckily for us, the pKa of this group is in the somewhat physiological range. So we reasoned that if we could use these lipids here, we could lower the pH a little bit during reconstitution, and it would be positively charged, which would help us to orient the BO3 oxidase. But then running the experiment at 8.5, which would remove the positive charge again. We confirmed that by measuring the zeta potential of these different liposomes, and you can only look at that at the last 3 rows. So you see here, this is the constantly positively charged, then DAP at 6.5 and DAP at pH 8.5, showing nicely that this is actually true. And now comes the strategy that we followed. So what we were thinking is, okay, let's make these liposomes. with 70% of the phosphatidylcholine and 30% of this DAP lipid here. We would lower the pH so our surface get positively charged. We reconstitute our enzymes. And now the trick to make the surface negatively charged, we would actually take advantage of the knowledge that we had that you can fuse liposomes if they are oppositely charged. So what we were doing is here, we were adding small liposomes. They were empty, but they were having a high content of negatively charged lipids. So after the fusion process, we can assume there has been a one-to-one fusion, and so overall the charge would be neutral. But if we now elevated the pH again from 6.5 to 8.5, the DAP would be no longer positively charged, and the negatively charge of the DOPG that we have inserted via fusion would prevail, rendering our final liposomes negatively charged, which should then be okay to accept the electrons from NDH2. And this is the outcome. I walk you briefly through that. Here we see our standard control. This is the synthetic system that I described to in the beginning, PC, DTT. And you see, if we only have PC and the membrane quinone, then the addition of NDH2 will not do anything. The next columns are these liposomes here. And you see, if we don't fuse anything with it, so we leave the liposomes as they are, then addition of NADH doesn't help. But if we make this step here by fusion of PG or PS liposomes, we would get back the activity that we already had with the synthetic system, showing us that we indeed can accept electrons from NADH, which is a more physiological substrate. And interestingly enough, if we added some cardiolipin into these liposomes, actually we were able to reach quite good yields in this production. So you see, these are two completely different strategies on how you might be able to orient the insertion or the orientation of membrane proteins into liposomes. And a last short bit on this first part is our recent efforts to produce ATP by electricity. Now, there has been a very nice work a couple of years ago from Tobias Erb's lab, where they have used electricity to exactly do that. So they have run a neat little substrate level phosphorylation cycle where they would add in the electrons on this side here, and they would deposit them on NADPA, on NAD plus, and create NADPH. And one of the additional steps would also create ATP. So this was very elegant, but it came with the limitation that it has to run under anaerobic condition. And this was for two reasons. So first of all, one of the enzymes was relatively sensitive towards oxygen, but then also the potential they required was roughly minus 0.6 volt against the standard hydrogen electrode, which means in the presence of oxygen, you are very likely to form reactive oxygen species. And then we were wondering if we maybe can do this with our system under aerobic conditions as well. And we were looking at our different entry points that we had for making our chemiosmotic coupling work. And NADH, which is the one that I just talked to you about, also has a relatively low potential. So we did not bother trying to do this, but instead moved in to ubiquinone. But we gave this up because we thought using a small chain, a short chain ubiquinone is not a very elegant solution, but we would rather have to have something that is more sustainable and maybe also more affordable. So we then ended up thinking about cytochrome C. And cytochrome C is, of course, the natural electron, the natural final electron donor in mitochondria. It gives 4 electrons to cytochrome C oxidase, which will then produce water from oxygen. However, the use of an enzyme as an electron donor comes with an engineering caveat, because if we look at the assembly that we need here, which is the AA3 oxidase, the cytochrome C has to be on the inside of the liposomes. Now, that's not too hard. We can get relatively easily cytochrome inside the liposomes. The problem is to get the electrons across the membrane. And we actually found out already earlier that this can be achieved by the electron mediator called PMS, which stands for phenazine metal sulfate. This is relatively easily reduced, for instance, by ascorbate, but also by other means. Now, what is so special about PMS? Now, PMS is this molecule, and there is not much detail you have to look at, but it can exist in three forms. It comes as an oxidized, a radical form, or a reduced form. So it can take up 2 electrons, but it's very willing to give single electrons away to reduce cytochrome C. Now, what is special about this is even if it's in its oxidized form and it's positively charged, it's still membrane permeable because it has such an elegant electron delocalization system. So what we were anticipating back then when we saw that this work is that ascorbate will reduce PMS on the outside, and of course in this very neutral form it will easily enter to the inside of the liposome, reduce cytochrome C, and eventually become oxidized. And what I think is very elegant about that system is that not only is this membrane permeable, but because this pump here will create an electrical potential that is positive on the inside and negative on the outside, it will actually drive the efflux of the oxidized form by the potential. And we were thinking maybe we can take advantage of this also to drive our system from pure electricity instead of ascorbate. And so this is the design we came up. We have separated the electrodes in 2 chambers, and we were hoping that our mediator would be reduced and the cathode would enter into the liposomes, reduce cytochrome C, and then start the reaction cycle. And to be sure that we can do this in a range where still no reaction with oxygen takes place, we characterize the system. And as you can see here in the cyclic voltammetrograms between the anaerobic and the aerobic system, the reaction with oxygen starts exactly at minus 0.6. So we had this window as long as we would be above minus 0.5 volts, then we should be fine. So we then titrated, or we did a cyclic voltammogram of PMS, and we saw indeed that we were able to reduce this in a range around minus 0.2, which still should be safe to prevent the reaction of the electrode directly with oxygen. We have 2 variants here, the classical PMS and one with an additional methoxy group. We haven't finished the system yet, but I think we tried it to show that it might actually work. And so this is just, we follow the reduction of cytochrome C via spectroscopy. So there is no ATP synthase involved, either free when it's just added to the outside of the cathode, but also when it's encapsulated within the liposomes. And you can see that the kinetics are exactly the same, indicating that there is really free diffusion of the electrons across the membrane. And if we do this with liposomes that carry an ATP synthase, we then also can readily see that if we apply a potential of minus 0.16 volt, that we can create a linear amount of ATP. Now, the yield we are getting, of course, is not yet at the level that we can achieve if we use chemical substrate. But then, on the other hand, we could also verify that this system is not producing any reactive oxygen species. So it might be a suitable system also for aerobic synthetic cells. So to sum up, this first part is, I think we can create these ATP producing modules from a variety of enzyme combinations. Many substrates can serve as initial electron source, and I think next to light, especially electricity or a hydrogen gas-driven system might be promising as they produce no accumulating product. Or in the case of hydrogen, the product that is actually produced is water. So with this system, we are currently able to get rates of several micromolars ATP per minute when we don't use too many liposomes. And so we can reach 100 micromolar certainly within 30 minutes or so. So I think we are reaching levels where this might become indeed an alternative to classical ATP regenerating systems. There is still a lot of challenges. So still there, you have to find optimal conditions for the core reconstitutions. These liposomes are okay for a couple of hours. However, we don't still know how their stability will be these under maybe more challenging conditions. And one aspect we are currently working on is to verify that we really indeed get a good efficiency between the redox reaction and the ATP production. And this includes to improve the tightness so that we really have high coupling and no loss due to uncoupling. So for the second, much shorter part, don't worry. I briefly like to talk a bit what we have been doing with GUVs. So when I came to Bern, I thought in 5 years from now, all the research I will be doing will be under the microscope. It's so great. I can see what is exactly happening. I can label my proteins. I see how many proteins I have into the GUV. I will use much less protein because there is so much less surface required for microscope. I will be able to distinguish between good and bad vesicles, and it will be great to test all kinds of membrane interactions. Well, it turned out it is great to test a lot of things, but it is actually quite challenging to measure protein activity in our hands, at least these type of activities that we try to measure. Challenges, of course, is heterogeneity of GUVs after production. Membrane protein insertion, I think there is a lot of protocols out there which might be very specific for their protein. I still think we are lacking general protocols for these issues, and we also don't probably have the solution. And there is many different techniques to produce GUVs, and I don't think that this helps the field a lot. So since we have to add things to the GUV, what we need is after formation, we have to immobilize them. We do this by this classical way using streptavidin and biotin. So we really coat them to the surface of the microscope slide. And we have found that you have to be careful with how much streptavidin you're using or how much salt you use. You can see that these liposomes really can sit quite tightly on the surface and make a very large interaction surface. And we are pretty sure that these tight angles here at the bottom is not ideal for the tightness. But it looks like otherwise our GUVs are quite tight, so they can keep a pH gradient. We don't know about electrical gradients, but otherwise they can keep gradients quite well. So in terms of incorporating membrane proteins, at some point we decided a little bit on to focus on this insertion via liposome fusion. So this is something we already did a couple of years ago, where we found that actually if we put one enzyme in a positively charged liposome and another enzyme in a negatively charged, the two would actually fuse completely, and we would have a core reconstitution of two enzymes. That was, of course, our vision on how we could create an endless number of proteins, different proteins into the same liposomes. And in essence, I think this works. We put the small liposomes into the positively charged liposomes. And when you see here, the GUVs are with no color, we then see as soon as we add the SUVs, which have a lipid color, we see that they immediately coat onto the surface of the GUV. And if you wait long enough, we think full fusion takes place although there is still, I think, quite a heating debate if maybe also sometimes just hemifusion occurs. This also works for protein, and that's also why we thought it is an elegant method, because you could kind of predict on how much proteins you will end up with by just having a different amount of liposomes added to your system. So here, for instance, we added three different amount of liposomes, and you can see that the amount of protein incorporated correlated very well with the amount of liposomes that we added to this GUE. So, and I will now present you data where it worked, but I want to be upfront, it doesn't always work like that. So the enzyme that we have been using here is cytochrome C oxidase, the one that I just described you in the electrical system for driving ATP synthesis with electricity. And here we drove the system the other way around. So we didn't have the cytochrome C on the inside, but on the outside, so we can add it easier. And this would lead to an actual efflux of protons from the inside to the outside, meaning that the pH will increase. And we tested that with pyranine, as many others do as well. The advantage is that this is a ratiometric dye, and if you build the ratio of the two wavelengths, you can get very nice traces. So you can see here, by addition of ascorbate and cytochrome C, we will see a very nice efflux, which eventually comes to a halt. And if we then add a pore where protons can flow in back again, like gramicidin, we see that this pH gradient drops again, and we get close to the initial value. So this is raw data that we get from bulk measurements as we have done it. And we were wondering, can we do this also under the microscope? And this is the raw data from one of these experiments. And you see that we roughly have like 20 GUVs within the same field of view. This is really something that we wanted to achieve, that we can observe the same reaction in the same field of view at the same time in multiple GUVs. And when we then extract the data, we see that there is slight changes, of course, in intensity, but of course it required the extraction of the data. And if we extracted the data of these 4 points here, now we can see actually that we were able to get exactly similarly looking proton pumping data as we were getting in bulk measurements. And of course, in essence, this is what I would like to achieve. And thanks to microscopy, we could then also analyze how these GUVs look like. And you see even this one, which almost looks like a pyramid. Still, there was a little bit of activity going on, although the gradient form was much smaller than the one in these most healthier looking ones. And this is then the data that we could extract from one single field of view. And this allowed us to do this very nice representation, and the system behaved exactly as we wanted it to do. So in a nutshell, I think in our hands, what we prefer now most over platinum coated GUE formation is PVA assisted, where we use polyvinyl alcohol. And we are also very happy with the yields that we are getting, and I also think we are quite happy with the homogeneity and how they look. We see that protein reconstitution via charge-mediated fusion works, and it fulfills our principle that we actually can titrate the amount of protein that we want to have in. But I'm completely honest with you, it just doesn't always work. And we tried hard to understand this, and but we are not at the level where we can say this is the way you have to do it. And that, of course, keeps it to be a generally applicable process. Also, from the work that we have done, I think we think GUVs might not be suitable to keep up high membrane potentials, and might therefore be limited to serve as bioenergetic membranes to convert proton gradients into ATP. And maybe this is also a lesson to learn from what we see in cells, where the cells, the big cells, they have plasma membranes which only keep a low membrane potential, while the high potential and the energy converting membranes, they are kept in much smaller structures like mitochondria and chloroplasts, or essentially bacteria. And then one other drawback that we are having is we can entrap small molecules quite easily and dyes. However, we are essentially completely unable to incorporate soluble proteins using either of these two methods, the platinum um, uh, electrode or the PVA assisted. So I think this limits very much the use of our efforts here. So the vision, nevertheless, that we are having is that GUVs might be suitable as container for synthetic cells, similar maybe more think of it as a plasma membrane and less as a really active membranes. We still will require to put proteins in there so that we can guarantee selective import and export of nutrients. I don't think that alpha hemolysin is the solution to our problems. But we have to be aware that probably we can only keep a low membrane potential. So this is something that has to be found out. However, to create energy and to regenerate energy, this must happen inside of the GUVs. And I think that's where our energy converting liposomes could play a role, serving kind of as mini mitochondria. We just have to make sure that there is a constant supply of substrate and removal of product necessary once we are within these vesicles, so they really can run for a bit longer than just a couple of minutes. And in this regard, I think membrane permeable substrates such as gases or light, of course, might be suitable. And I think we have these liposomes that can produce ATP from either of these substrates. But then, as the really grand challenge that I'm seeing is not to produce ATP. We can do that, but the really grand challenge is to bring the system out of equilibrium or a little bit closer to what we see in nature. In nature, we have high ATP to ADP ratio, which makes it even harder to produce the ATP. Then also, in the cell, we have a high NADPH to NADP plus ratio, but a low NADH to NAD plus ratio. And to find enzymes that actually are able to mimic this situation so that we can bring at least the basic energetics of a synthetic cell close to physiological range is something that is at the core, and I think it's also close to our heart, and it's something I would like to work towards in the next couple of years. I would like to leave you with a couple of very recent data where we make the first steps to put these SUVs into GUVs, and we faced that we would have to switch methodology, and so we teamed up with a team from ETH Zurich, Stavros Stavrakis, and his PhD student Chow, and they essentially copied for us the octanol assisted GUV formation. And this is how the picture looks. So we get the SUVs into these GUVs, and what we were facing as a problem when we were doing this is it was relatively easy to get these GUVs, these good-looking GUVs, as you see, they are. I thought this was a movie. It seems not to be a movie. Oh no, I think I have to. If I remove this, I think it's a movie. Yes, so we get a very good yield. I think they with optimization of the design, they could increase the rate of formation. But we saw that a substantial amount of octanol was left in the bilayer, and we've found that this would prevent, for instance, proton leakage. We also found that this would prevention of fusion with liposomes, and we had then to do an additional dewetting step to get rid of the remainder of the octanol. But eventually we were then able to create really GUVs that were both containing cargo on the inside, but also proton on the outside. And What has Larissa been doing, a PhD student in my lab? She tried to see how easy is it to load these QVs with different amount of small unimolecular vesicles as they might contain the energy converting enzymes. And you see here the range of zero point 0.5 to 10, which is relatively high. And I also on purpose brought you images where it doesn't look so good. So you see we are facing a lot of problems. We form aggregates. Sometimes the yield is quite poor. And we find there is a lot of impact on the buffer composition. Ions, especially magnesium and phosphate are a burden, but both of them are required, of course, to produce ATP. And to regenerate ATP, we need phosphate and also magnesium for binding. And then we also found that the osmotic balance is really something that we might have underestimated a bit. But Larissa is doing great, and she also tries to follow our initial intention to not just look at single QVs, but really look at them on a more global level. And she put some effort in using good segmentation methods that would allow us to extract the data. In a nutshell, this is very recent data and really not made up, really not brought to the latest level, but you can see clearly there is a trend that the more SUVs we add, the more is also really incorporated. So this gives us some hope for the future, and that we indeed soon might be able to have this minimal mitochondria within these QVs, and then we can create the ATP within QVs on demand. With that, I would like to acknowledge the people that have been working on this. So this has been also work from previous PhD students, Andrea Amati, Stefan, and Sabina Deutschmann. Stefan Morning is finishing now. Nicolas has also finished, and Larissa and Ping, of whom I haven't shown data, but also works on the project, are still ongoing. Then I would like to thank Peter Bröckmann from our institute, who knows much more about electrochemistry than I do, and I would like to acknowledge collaboration with ETH Zurich and University of Freiburg, Thorsten Friedlich's lab, that helps us with the purification of these oxidases. And I would like to thank you for your attention.
Thank you. Thank you so much. That was really fantastic, especially for me being a liposome person. It's nice to see someone talk about liposomes actually realistically acknowledging the difficulties of that system.
Yes.
There's a lot of questions in chat. Can you see chat?
Yes, I can see that.
Can you read, if you read a question out loud before answering, that's going to help people watching the recording because they don't see chat.
Good. Does the hydrogen consumption change the pH? Does it affect liposome stability if pH changes? Now, I assume you refer to the study where we have used hydrogen. So, as a matter of, or let's put it like that, if you refer to this third option on how to create a proton motive force that I have displayed next to bacteriorhodopsin and the redox pump in this introductory slide, yes, it changed the pH. That's actually the purpose on doing it. So they oxidize so much hydrogen that it actually releases a lot of proton, acidifies the outside, which would then eventually drive ATP synthesis for a limited amount of time. Now, if we talk about the hydrogen consumption that we used to drive, okay, that was the one I was asking about. Thank you. Good, then I can switch it. But it wouldn't change the concentration, the pH in our system, where much lower concentrations were used. So a second question is, why did you choose to model your ATP synthase system pumping out ATP outside of the cell rather than inside? How do you preserve energy in this situation? Um, so, so I think it's a very good question, and the honest answer is, of course, there is not so much that you actually can do to correct to orient the ATP synthase that it looks with the headpiece to the inside. And I think this is its original orientation, but as you might know, if you break cells, they tend to form inverted vesicles. So, and I think somehow the ATP synthase knows that maybe I'd rather have to a little bit more space, so it keeps the head on the outside. But then we also found that we can control reconstitution much more if we use this method where we just saturate the liposomes with detergent, because then they don't break and we have somehow this control that hydrophilic head groups like the F1 group stays on the outside. We think also, this is not a disadvantage because since we just use this internal container as a proton exchanging system, and despite lacking a protective membrane, I think it is a very similar situation as we will find it in mitochondria. Now, of course, in mitochondria, ATP is produced in the matrix and is exported via the ADP-ATP exchanger. And I think Bert Polman's lab has shown that you can reconstitute the ADP-ATP transporter. However, I think for right now, this is not something that we have to worry too much about. Does the size of the liposome, so membrane curvature, matters for the insertion efficiency? It's a very good question. The thing is, the insertion of a liposome is an extremely poorly understood process, and I could easily go on this question for a very long time. But in a nutshell, what we are seeing is that as long as we keep the size of the liposome, let's say between 50 and 200, we don't see much of a difference. So we were hoping that if we would use very small liposomes, then the internal volume gets very small compared to the surface. And we then thought, okay, then we might get much more rapid acidification. We might create higher PMFs, but obviously this comes at the cost which we don't fully understand. But if we take the same number of proteins and put it into 50, 100, and 200 nanometer liposomes, there is not much difference, and especially not a very reproducible difference between the size of the liposomes. So I think for the insertion itself, the curvature itself does not matter. Still, these are relatively small proteins when it comes. This might be different playing around with larger proteins, maybe like complex 1, but I think for bacterial enzymes, this is not a limitation. Then I have one from Hillary. This might be, there is no naive question, but when you use electrodes in presence of liposomes, how do you avoid disruption of liposomes? I thought electric field is a method of liposome formation, so it affects charged lipids. Apologies if this question shows some large knowledge gap on. No, I think it's a very good question, but I think you're probably talking about two different things. So when you use electrical fields for liposome formation, you don't deposit liposomes, or at least we didn't depot the liposomes on the surface, but actually lipids. So the lipids were then deposited on the electrode surface via organic solvents that would dissolve. So the potential at the electrode would actually only help the hydration process of these liposomes. But this would happen before actual liposome formation. Now, if these electrons might harm our already formed liposomes, I don't know, but I don't get the impression that this is happening. I think these liposomes are relatively stable, and there is very nice data from Lars Jeuken's group where he actually shows that he's even able to directly deliver electrons onto the Q10 that is within the liposomes. And we were also thinking of doing that because that would avoid the problem of having to have mediators. But then the problem is, of course, that we limit ourselves to the surface of the electrode, because then the remainder of the solution has to be essentially free of liposomes. And we just use the electrons to reduce the mediator, and we stir our solution so that we can take the full advantage of the 3D situation of a stirred solution. How light-driven transformation of your chemically prepared system can guide the development of primitive photochemical modules capable of self-sustaining metabolic cycling. Said, "Oi, this is a lot of complicated words. I'm afraid I might be a bit tired to fully understand that." Um,
Hello, I'm sorry for
so
that, but we have a
maybe would you be able to explain it to me maybe in simpler words?
the thing is that we are working on a biogenically synthesized system. It
Yes.
is prepared from a mixture of inorganic organic substances,
Yes.
and it has a redox system.
Mm-hmm.
And we have redoxins for electron transfer
Mm-hmm.
and those things, and decarboxylation reaction as well,
Mm-hmm.
and a membrane for charge separation.
Mm-hmm.
So they mediate the photocatalytic process. They
Mm-hmm.
can photochemically fix the atmospheric nitrogen into fixed nitrogen
Mm-hmm.
and atmospheric carbon dioxide into organic carbon.
Yes.
Possibly the molybdopyridoxins in the mixture, they're playing a great role.
Mm-hmm.
And I was working with Hall in UK, Professor T.O. Hall, and he studied actually detected the presence of redoxins in this mixture.
Mm-hmm.
So we have the photophosphorylation reaction in the mixture. We have the mechanisms for electron transfer.
Mm-hmm.
I need to characterize basically the outer membrane uh, of these systems to further look into this mechanism.
Mhm. Okay, I
So
see. Yes,
yes,
yeah. I think it's very interesting, and I think it's one of the challenging ways. Nature is able to combine all these different systems so elegantly, and it's somehow managed that they don't interfere with each other. But we
yes,
humans, we just understand a little bit of it, and we play around with electron mediators, and of course they go everywhere. But speaking of nitrogen fixation is really interesting, because I think it's also one of the process where you both need redox power and ATP. So ATP could, in this sense, provided by, for instance, by a light-driven transformation, which would not affect the redox processes of itself.
yes,
I
yes,
think one of the trouble is really that, in synthetic biology, when we mimic this system, that we don't interfere too much. I think this is the price of evolution, that they found solutions so they don't they don't mess up each other's systems.
yes. I will share
And
one of paper of my lab
yes.
where this presence of nitrogenase, and they have passed acetylene overhead the mixture, and it was reduced to ethylene and finally to ethane. And the
Mhm.
hydrogen is coming. We used radioactive heavy water, and the hydrogen is coming by the photon decomposition of water.
Mhm.
So we are having a photocatalytic system which can take energy from sunlight and
Okay.
decompose water into hydrogen and oxygen.
Yes,
But
yes, that's.
yes, so we have the basic things, but we need further some more characterization. And
Mhm.
I will be in touch with you, sir. And let us see that I will seek your guidance in the characterization of the membrane of
Yeah,
this
well,
system.
we can keep talking after that. Thank you.
Thank you very much.
Yes. So the second question is about methyl biology. I don't have any experience. I know it's very
Okay,
widely used, but
it's pretty good
I
one.
think it's very strong, and we typically try to stay in the range where we don't need methyl
Correct,
biology.
correct, sir.
And then the last question is, for synthetic cell applications, how to avoid liposome fusion to make bigger cells. And so This is a very good question. So, if not absolutely necessary, stay away from positively charged lipids. They really do a lot of things. They fuse. So I think you have to somehow saturate them. So you always have to have more negatively charged lipids so that you can make a controlled fusion step. You know that you say now I'm doing membrane fusion, but after that fusion stops. And we were also looking at other ways on how to do fusion, for instance, with snare proteins. And that in principle worked very well, but it's of course much more cumbersome. And other approaches to fuse, that was really tricky. So the approach that we used for liposome fusion to orient that, I think, was really special, and we were just glad to have found a solution to that. And for bringing proteins into GV, I think in principle it is a good method, but there is really a step that we don't fully understand why these GUVs don't work better when we look at them during activity. They look fantastic under the microscope, but when we look at that activity, they are just mediocre, and it's really something that can keep me awake for a very long time at night.
Kate, can I follow up
Yeah.
Yes,
question? So thank you so much for the answer. The reason I'm asking that question is I'm not a synthetic cell person, but my colleague doing a lot of synthetic cell work with the
yes.
Kate Guatemala. I actually use liposome for another application. I want fusion actually.
Uh-huh.
The
Okay.
reason is I'm using liposome for the drug delivery or DNA delivery
Mm-hmm.
or whatever nucleotide delivery. And then we found uh, it's very good.
Mm-hmm.
Not that efficient compared to other methods, but still sufficiently
Mm-hmm.
efficient, uh, even both gram-positive and gram-negative bacteria.
Mm-hmm.
But in the literature, you know, it worked for both gram-positive and gram-negative bacteria, but somehow some people have some, uh, I would say, some wrong kind of perception. The gram-positive bacteria is much more challenging to have fusion and then delivery the DNA and etc. Is it true, or I just want to understand why
Well,
people have that kind of
I, I,
perception?
I would think that for grandpas, well, yeah, I studied, I did my PhD in the Institute of Microbiology, but I don't know so much about it. But I would think that it's the cell wall of gram-positive bacteria that is seen as a physical hindrance that they actually reach the part of the membrane where you can see fusion. That you don't even go there. I think if the polar, we have done very fun experiments. So we have shown that we have produced bacteria. It was just membranes of bacteria. I admit it was not living bacteria, but we were able to actually fuse a protein that we have purified into existing bacteria, and then it would work. So I think if you have enough surface and if you have enough polarity of these liposomes, I think fusion can take place. We have seen that there's curvature plays a role. So small liposome tend to fuse much quicker, probably because they can release the curvature stress. Also, the sheer percentage of charged parts plays a role. So if you have a large proportion of charged lipids, they fuse faster. The presence of salts makes a difference. Um, if you want to pass the cell wall of gram-positive bacteria, I think in the beginning it is mainly a physical problem to overcome.
Because the gram-positives have the thicker,
Yes, yes.
whatever the
They have a
barrier.
very thick wall with a lot of sugars, and most things just don't get into the region where they would be able to actually get to the plasma membrane.
Okay. Are we able to make the DNA delivery through the gram-positive and gram-negative as well. So I do not have any problem, but the people,
Okay,
criticize our actual proposals. I just wondering what's the people's perception in the
Yes.
field, because I'm not the
Okay.
expert in the liposome, but I'm using liposome and then optimizing the liposome the
Okay, yes.
condition. But it worked. But the academia, a lot of people has their own idea, and then if they believe this is the case, they just kind of giving a lot of nonsense kind of criticism.
It's
I do not appreciate it. So that's why I'm asking you, because you are the expert, I guess. So I now
but
feel better because I could write back saying, the reviewer, you are wrong. I can do that. Thank you.
what they say of the field of bioenergetics is that it's strong personalities but weak data. So so
Okay,
yes,
thank
to
you.
change one's opinion, yes. Good.
Thank you. Thank you so much for answering all the questions. Thank you for a fantastic discussion. And I'm there too. I'm really looking forward to seeing where that goes, because we need this system. We really need it to work for a synthetic cell to do stuff, to do useful stuff.
Yes, I think it is really something that we can't just leave out. I think we need the energy.
All right. Thank you so much. And thanks everyone for a great discussion, and have a wonderful rest of your Monday.
Thank you.
Thank you very much. Thank you for having me.
Great talk, Christoph.
Bye.
Thank you. Thank you.
Okay.